I'm going to talk about, motivate with a little bit of background, just a wee bit, and I'll kind of infuse background as we go through the talk. And then, question? You guys have a question? Okay. It's okay. And then we'll go through the theory of SHG, and then I'll talk about SHG in contrast to two-photon fluorescence microscopy. All right, so I know you guys are, this is a microscopy class, right? So you've covered quite a bit about the fundamentals of microscopy. So some of these things you'll recognize, some of the things you may not recognize. Generally, I like to throw up a Venn diagram of sorts that kind of tries to explain what the overarching theory that one could invoke to be able to describe the phenomenon that's under investigation. So for something that is simple focusing, you know by now that you can explain it strictly using ray optics, correct? For the work that we will discuss today, it's sort of a hodgepodge of quantum optics, only in terms of the language that's used at some point, but not any real hardcore quantum mechanics, and mostly electromagnetic optics, right? So electromagnetic optics is what we know as being the most complete theory of optics that does not have anything to do with quantum mechanics. Okay, so it's almost there in terms of being able to explain pretty much a lot that we normally observe in optics, including microscopy. So, source material. Some of the materials from, a lot of the materials from this book, and or this book. If you're a microscopy person or you're interested in learning about microscopy, I encourage you to especially get this book, Pauli. Right? It's considered the book. There's another book I didn't throw up there by Lakowitz on fluorescence. All the chemists, all the optics people usually have that on their shelf somewhere. And this is quite a nice book too. Even though it says confocal and this says confocal, they cover a lot of different things that's related to microscopy. So if you're interested, if you want to learn about fluorophores and things of that sort, I recommend you to get these.
What was the name of fluorescence?
I think it's called Principles of Fluorescence Microscopy, Lakowitz. Right, so a lot of the chemistry students, physics students, they typically deal with fluorescence or fluorescence energy transfer. They would have these books. So I encourage you guys to also pick it up. So, some things to note. Let me see if I can move around a little bit. Okay, some things to note. For the background is, this is all about light-matter interaction. I'm focusing on the nonlinear aspects. Now I'll tell you what I mean by nonlinear as we go on. But some key points to keep in mind is that light interaction with matter is mediated through electric charges. Okay, electrons zipping around, right? And you have an oscillating electric field, and one often invokes something like a Lorentz model, for example, to describe some of the phenomena that they observe. Time-varying electric fields, like what you have with light, exert forces on atoms, molecules, and solids. So the molecules comprises the atoms, and the solids comprises the molecules, right? The interaction is governed fundamentally by the rules of quantum mechanics. It's just that you don't necessarily need quantum mechanics to describe everything, right? So if you want to just describe regular reflection phenomena, focus of phenomena, you don't necessarily need quantum mechanics any more than you need to just use it to describe a ball rolling down a hill. You can get away with just regular Newtonian physics, right? Vibrating electric charges can absorb and emit light. So that's a key point in understanding light-matter interaction, is because of these transitions from lower energy to higher energy, from higher energy to lower energy, is how the medium interacts with the environment, okay? Now, when we talk about linear optics, which is everything that I'm sure you all have learned, at least up until this point, there are a couple of things that I like to note. First, the properties, and specifically the optical properties, which in general could be complex, right? So complex properties usually imply something about the medium being absorptive, right? So the index of refraction, all your optical constants become complex functions, right? And so your index of refraction becomes complex. It's a general expression. We now use the language of dispersion. And so your real and imaginary parts of the index of refraction are independent of the light intensity. That means if I take my laser pointer, stick it through a piece of glass, and if there was a knob on this, and I can crank up the intensity of the laser pointer, I don't expect N or alpha to change as I crank the intensity up for a linear medium. Okay? The principle of superposition applies. You're engineers, specifically electrical engineers, so you know you've done classes in systems analysis, right? And you know that linearity is one of the hallmarks of engineering. We try to make things linear, force them to be linear, so that now we can handle systems or interconnected systems or multiple input, multiple output systems. And the principle of superposition is at the heart of that. It's no different with optics. So this is optics really coming from an engineering perspective in terms of how we're teaching it. The frequency of light does not change from passing through a medium. Again, if I'm walking down the street and I start shining a laser pointer, something I'll never do, of course, and if I shine it through the air, see that green laser pointer remains green, right? But with anything nonlinear, right? Whether you're talking about a harmonic oscillator, mass spring system, something acoustic, something optical, the moment you say nonlinearity, that means you're also adding additional frequencies into the system. Okay? For us, the additional frequencies would mean color changes with the laser light. Okay? And so you hold that thought in mind. And two independent beams of light in the same region do not interact with each other. So we say the light cannot control light. So in other words, if I take two laser pointers, shine them through the air, and I start flickering one laser pointer, I don't expect it to affect the other laser beam at all. Right? All hallmarks of a linear medium. So you can deduce what the nonlinear medium will be, right? It's kind of like the opposite of everything. The refractive index will depend on the optical intensity. And by the way, it would be torture for you to copy every slide. Okay, so that'll be draconian. So what I will do, I will email Professor Popescu these slides once I get back to my office, so that you can have them. Okay, so relax. You know, just maybe take some shorthand, but don't try to copy everything. Okay. The principle of superposition does not apply in a nonlinear optical medium. The optical frequency changes as it goes through the medium, so I can send in the green light, but I may also get something in the UV at the output, for instance. And two independent beams of light can interact within a nonlinear medium, and we'll talk about how, okay, because essentially the medium acts as a proxy for mediating that interaction. So one can ask the question, where is nonlinearity? Suffice to say it's not in the light, it's in the medium. And you pretty much can make anything nonlinear if the intensity of the light is strong enough, right? Or the intensity of the electric field is strong enough, right? So the medium changes its properties somehow based on the light field interaction with it, which then modifies the refractive properties, or in some cases maybe the dispersive properties of that same medium, which of course is going to affect light propagation. Right? And that's all it is. Okay? So generally there are two pictures. One is the dipole picture, in which we can think about where the nonlinearity may be. The way I like to think about this, again, sort of mass spring. So you can think of a dipole. So you know you have some charge, right, surrounding some atom or something, and so you have an electronic cloud, and you apply an electric field, right? You're going to have charge displacement, correct? And that charge displacement we call a dipole, right? That has some potential, right? So what happens is one can imagine in a 1D case, you know, so you have some dipole moment, P, is equal to minus E, which is the charge, times the displacement X. Okay? Mass, spring, rigid body. We displace it, and within the linear regime, we can model that by Hooke's law. F equals minus Kx is a restoring force. Okay, it's no different with charges. Okay? Now, what happens if you start stretching that spring too much? It's no longer harmonic, it becomes anharmonic, right? And you start deviating from the linear regime, and you start introducing now additional frequency components. Okay, that's the same idea. Field is strong enough, that's charge displacement makes it harder for this charge to go back to its equilibrium position, right, in a linear way, right? So you're introducing now additional frequency components with nonlinearities. So the other way of thinking about it is that this is essentially also a quantum picture. Is that, let's use the language of atoms. The number of atoms that occupy the various energy levels depend on the photon flux density. So, you know, you have populations. You have populations in the ground state. Yeah, if you're thinking of electronic structure, for example, you have populations in the ground state. You have populations in an excited state, right? What are the number of atoms that are occupying this excited state? What are the number of atoms that are occupying the ground state? Okay, so it's going to depend also on the number of photons that are entering the mode. Okay, and that's where you can get nonlinearity from. Okay, and this shows up a lot in laser physics, of course, because that's how you explain stimulated emission, for example. Okay. Now, as engineers, we like to throw up sort of a systems picture. P here, we know from electromagnetics, that's the polarization density. So that's taking all these different dipole moments that are in a system and adding them up. Okay? So you have some that are this way, some are this way, some are this way, and you just sum up the entire thing over the volume. You're going to have some net polarity, if you will, some net dipole moment. So we call that the polarization density. And in general, within linear optics, and what you would have learned, P is proportional to E, right? So we know like epsilon naught is the free space permittivity, and chi is electric susceptibility. From this, we can derive the index of refraction. We can talk about Snell's law and all the things we will learn from general optics. Correct? So in a systems picture, we say we have some system that's linear, maybe linear shift invariant, for example. It's characterized by some chi. We apply a field E, and we have a response P, the polarization density. Okay. Hopefully you guys are following so far. All right. As we become nonlinear, this nice equation of a line starts to bend. And it bends as the field intensities become higher. Right? And as you guys would have learned in introductory calculus class, you know, if you have some nonlinearity in your curve, you can linearize it about some region. So in this case, what you do is you take a point, you take a tangent line to that curve, right? And you do a Taylor series approximation, grab the first couple of terms, right? This is where we talk about linearizing our function, right? So in this case, you're basically doing a Taylor series approximation about E equals 0, right? And what you now have is a term, the polarization density, in terms of higher order exponents of the electric field. Okay? So this is the first order term, second, third, and so forth. And these are associated coefficients that go with it. We're talking today about the Chi2 process because that's what we're interested in for the stuff that we do in my lab. And so all of this is nonlinear. Right? And the higher up you go, the more rare the process, the harder it is to achieve. So this work, this becomes important when the applied electric fields is comparable to the fields that are binding atoms together. And I give some estimate of what that is. And it wasn't until, sure.
Sorry, I don't have any background on this. So the exponents, 1, 2, 3. So
E squared, E cubed,
So then, in terms of the medium or what's happening, what does that mean?
So what happens is this, is that, so remember that restoring force picture? And I said, okay, this, you know, ideally this is all this nice line. When you take a field and you apply a field to your medium, if the field is strong enough that you apply, and it's strong enough relative to what? Strong enough relative to the fields that are keeping that medium together, right? So that you disturb it from being in equilibrium. Then this is no longer a line. This is bent. This is a curvy. But you want to work with it. And you want to be able to solve problems. So what we normally do is we try to linearize about some nonlinear function. So we look at where the nonlinear element is, and we may say we're going to draw some tangent to the curve. We pick a point, we call that the equilibrium point or the operating point, and then we linearize about that point, which is equivalent to taking a Taylor series approximation about E equals 0. And that's how you arrive at this. Any questions? Other questions? Okay. So this could not be observed because you didn't have fields that were strong enough relative to the interatomic fields to see that effect until the laser was born, right? Which was 1960. So the first observation of this was about 1961. Okay. It turns out it was exactly second harmonic generation. Okay. So what you do is a small perturbation. This is also called perturbation analysis around E equals 0. Your fields aren't quite this high, but they start to become high that you start to incrementally deviate from linearity. Right? Now all you need is a little bit. Quickly in optics class, you can't get away without throwing up the wave equation. All right, so this is a wave equation that you all have seen. Isotropic medium, except here now you see there is a source term. We'll say something about that in a second. So if the medium is nonlinear, homogeneous, and isotropic, you still have this expression for the wave equation, except now what we've done is we've taken for the polarization density, specifically the nonlinear term, that becomes our source term that generates a new field inside the system. Okay? I know you all talked about the Born approximation in probably a lot of detail for Professor Popescu's class. So I'll just say a couple of words about this. It's useful, not just for phase imaging, but for any type of light scattering processes. And the process that we will talk about today, this nonlinear optical scattering, it's scattering. Right? So to solve this, we take what's called the first Born approximation. So essentially you have some applied incident field. It's applied to a nonlinear medium that's over some small volume, for example. That field creates, vis-a-vis the nonlinear polarization density, it creates a new source term, which was S of E. And that source term now generates a new field. Now that new field can generate now a new source term, which generates a new E2. And you can keep going in this feedback loop. Okay? But if the scattering process is weak enough, then you basically are saying that you only have one scattering event, right? And we call that the first Born approximation. That will be sufficient to describe the generation of a new field. And what one does is basically use diffraction theory, new point source, and then you're going to have radiated multiple spherical waves, and then you add up their contributions in the far fields, and then you are done. Okay? So you have some very fascinating, I just put up a couple of interesting ones, or at least some more popular ones, some interesting consequences of nonlinearity. The first one, optical frequency conversion. Right? So what you have here is a nonlinear system, and you have at least 2 laser inputs to that nonlinear system. Okay? Omega 1, omega 2 correspond to the associated frequencies. And what does that nonlinear system do? Well, what it will do, this could be, for example, a Chi2 system. It will add those frequencies together, potentially, and you can subtract those frequencies together, potentially. When we add them, we call that sum frequency generation, right? So you have a higher frequency, lower wavelength, right? When you difference them, we call that difference frequency generation, okay? Lower frequency, longer wavelength, okay? And they have to satisfy what's called energy conservation. Omega 3 should equal omega 1 plus omega 2, because this is not about absorption. This is about some mechanism of scattering. And I'll show you that also in the quantum picture in a few minutes. You can put a filter here to block out the colors that you don't want and let the color that you do want through. Okay? With OPAs, and any of you who work in a lab, maybe with nonlinear optics, you would see this. Optical parametric amplifiers. It's an amplifier, just like you will learn in the circuit theory class, right? Nonlinear crystal, You have some signal that you're interested in amplifying. We call that omega 1. You have some seed. There's going to be a reservoir of energy. We call that a pump, given by omega 3. In the crystal, omega 1 draws energy through a nonlinear interaction from omega 3, so that omega 1 starts to become amplified, but it's coherently amplified. What do I mean by coherent amplification as opposed to incoherent amplification? Meaning that you're not only amplifying the amplitude, but you're preserving the phase relationship. Okay, again, that's something you guys would have learned, right? And so now you also have this auxiliary field that's generated. We call that the idler. That just means we don't use it. Okay? And then you can block out the stuff that you don't want. You let omega 1 amplify through. That's an OPA. Or you can have another one called an optical parametric oscillator. All right? And so what's an oscillator? So you put it inside a light box. Light boxes, we call them optical resonators. Right? Pair of mirrors you slap around each one of these crystals. Now all you need to do, so each mirror is not perfect, is almost perfect, high 90s percentage in terms of reflection, right? Just like a laser, right? And you let a pump beam through, excites this nonlinear crystal, so you start generating these other frequency components, omega 1 and omega 2, but most of the light's going to reflect back, and that process is going to go back and forth over and over and over and over again. So all of a sudden now what you're doing is you're generating additional new frequencies at wavelengths that you may sometimes not even have access to, which is why people use OPOs, right? So in my lab, we have some type of titanium sapphire laser. It goes from about 700 nanometers to a micron, but we would use an OPO to extend that bandwidth much further into the mid-IR, for example, by way of this example. And the final one, spontaneous parametric down conversion. This is used a lot in quantum optics. When I was a graduate student, I worked in quantum optics, so we use SPDCs as the workhorse all the time. And it's essentially what you have is a nonlinear crystal. So you basically have the OPO with no mirrors. Okay? So you have a pump, maybe in the blue. It pumps this crystal. Now, what the spontaneous parametric down conversion, all that means is that you're going to get multiple solutions to your omega 3 equals omega 1 plus omega 2, right? So you're going to get a ring of colors, okay? Sometimes you can get 2 rings based on the birefringence in the crystal. That's an additional story. And each one of these pairs of these photons, there has a corresponding sister photon that we say is entangled with it. And this is how people do quantum teleportation, quantum cryptography. This is a convenient source. So you can use the same nonlinear crystal that you do use to get second harmonic generation, beta barium borate, for example, BBO, except you pump it differently, right? Pumping it out with, let's say, blue light as opposed to pumping it with red light, and you're going to get some weak distribution of a ring of colors. It's kind of cool. You can actually see that ring safely with goggles, okay? Questions so far? It's a very, very quick primer, okay? So we're interested in second order nonlinear optics because we're focusing on second harmonic generation. It's a second order process, but it's actually called 3-wave mixing because you have the pump wave as well as the generated wave that participate in this. And 2 waves of frequency omega 1 and omega 2 can interact in the Chi2 system to generate a 3rd wave or a 3rd frequency. Unlike fluorescence, have you guys studied fluorescence at all? A little bit, okay, I'll say a little bit more about that. So unlike fluorescence, this is not molecular absorption, okay? If anything, it's molecular scattering, okay? And I'll tell you the distinction in a few slides. So the chemists like to use a picture like this sometimes. It's something called HYPER-RAYLEIGH scattering. You guys study Rayleigh scattering?
So we just started on the Born approximation. We have more
Okay.
down to
Okay, no problem. So Rayleigh scattering, all that means is that it's the regular scattering you expect. Have the wall shine light off of this. There's a weak component of green shining off of the wall, right? So the green light a little bit scatters off of that, or just shine a red light on a mirror or something, and that comes out, okay? So the frequency that you put in is the frequency that you get out with Rayleigh scattering. And for certain types of molecular molecules or arrangements of molecules, you can have a distribution that has some point group symmetry that is considered to be non-central symmetric. So this has to do with the distribution of atoms within a material. Okay? And if you have atoms all over the place and you have one looking down through some plane, if I flip that plane upside down, do I have another atom in this twin place? If it does exist there, we call that central symmetric. That means there's a point of inversion symmetry. Okay? If it doesn't, we call that non-central symmetric. Chi2 systems, for that process to exist, have to be non-central symmetric. So at a molecular level, that means anything that's chiral. Okay, chiral would be like, like DNA is chiral. Myosin, muscle fiber, is chiral. Okay, certain types of polar molecules, you can think about chirality. Yep.
And that's in the macromolecule, not in the individual atom.
That's right.
Yeah.
That's in the macromolecule. That's exactly correct. Yep, that's correct. So what happens now with a non-central symmetric structure, if I shine a wave at a frequency of omega onto it, there's something that breaks that symmetry. I'm going to get that red light back out, but I'm also going to get an additional frequency component out. That's a 2 omega. Okay, if anyone's interested, actually, you also get a DC component, right, which leads to optical rectification, and some people also use this for terahertz generation. Okay, so that's called hyper-Rayleigh scattering. Now, one can imagine each one of these molecules, and you guys would appreciate this being electrical engineers, each one of these molecules can be oriented randomly in three-dimensional space, right? So from an antenna perspective, each one of these is radiating like an antenna, but is radiating effectively out of phase with respect to the other ones. So as a result, the net field that you record in the far field would be considered to be incoherent. That means it's not highly polarized and it's not highly directional at all. So a lot of chemists use this hyper-Rayleigh scattering in solution. Okay, so the individual process looks exactly like our Chi2 process, because it is a Chi2 process. But what you can measure, look, we can't see a single DNA, a single chiral molecule, but we look at collections of those. So we have some supramolecular order, and we look at how they are arranged with respect to each other ultimately, right? And that gives us some sense of the nature of our radiation pattern. Okay. So here, HRS becomes coherent if they're all aligned, if they're all in phase with respect to each other, right? And now they become highly directional, right? So this red light interacts with this distribution of molecules, and they're all oriented in the same way. They're all going to radiate in the same way, subdiffraction, you can't see it, right? And what happens is that now the radiation, let's say, is in the forward direction, and it's highly directional, meaning that automatically it's going to be highly polarized. Right? And that's when we can use the language of second harmonic generation. Just interesting. That's one of those things that the physical chemists like to talk about. Phase matching is something that is usually used to describe the conservation law. Somebody might be interested in saying, oh, that sounds like a really cool thing, second harmonic generation or something like that, but are you breaking any laws of physics? Obviously not. We are conserving energy, right? Our 2 photons, their energy, or their frequencies. You can, if you wanted this energy, is multiplied by h bar, of course, right? But their frequencies have to, the sum of their frequencies has to equal the total overall frequencies, right? So that's our omega-3. So in this case for the SHG, well, hold on for one more slide. And the momentum matching conditions says the same thing in terms of the directionality of the waves, right? So all these wave vectors, the K1 and the K2, their resultant should be K3. You shouldn't have something in some random direction, right? And so we call this phase matching. So with SHG, the only difference between SH, some frequency generation, taking two frequencies, adding them up, and coming out of the third frequency, becomes second harmonic generation when those two frequencies, omega 1 and omega 2, are equal, the so-called degenerate condition. And when they are equal, that's when we're talking about SHG, right? So omega 1 plus omega 1 equals omega 3, which equals 2 omega 1. So in other words, two input red photons at 800 nanometers interacts with the nonlinear crystal, lithium niobate, or BBO, and it generates one photon that's blue at 400 nanometers, half the wavelength, but twice the harmonic. Okay? You do the same idea, they stay at the same momentum, and you come up with a momentum matching condition. And there's such a thing as perfect phase matching, so this is all ideal. In other words, there's no residual when you add up in phase these vectors. And when you start to deviate from perfect matching, phase matching condition, you lose the ability to generate any signals, okay, in general. Okay, so if I gave you a nonlinear crystal, right, took you to a lab, gave you a laser beam, gave you a lens, focus into that nonlinear crystal, your red light will become blue. It's a really cool effect. You see it quite easily. Okay? Phase matching means the crystal is cut just the right way relative to the direction of the laser beam for that particular color of laser beam and for a particular polarization. Okay? If I rotate the crystal, my light at the output starts to dim. And I keep rotating, and eventually it goes out. Right? So you just don't get it anyhow. There has to be certain conservation laws. And that's what all this is about. Okay? But all of this is relaxed when you focus tightly, like in the microscope, and when you deal with biological tissues, because they're extremely heterogeneous. So the quantum picture, something like a Jablonski diagram. Here, so this is an energy level. Yep?
About your other example.
Yep.
What happens to the polar, like if you excite with a certain direction of the polarization, then the generated light will have the same polarization as the incident, or?
No, it's a good question. So what happens is that, so I'm skipping over certain things. So for something like second harmonic generation, you can have type 1 second harmonic generation, you can have type 2 second harmonic generation. It depends on the type of birefringence that you have in that crystal. And one leads to something where you have orthogonal polarizations, the other one leads to something where you have parallel polarizations. So it's not quite as simple as the way I'm making it sound, but the polarization is quite important. If your crystal is cut such that it really is only designed for one polarization, then you start to lose it when you start to deviate from being maximally parallel to a situation where you only get some projections of the right polarization onto that axis to one where you're completely orthogonal, and you're not going to get anything at the output. We'll talk about what happens when you focus. Because when you focus, you get momentum diversity, and so that relaxes things. So here, what you have is a lower energy level, ground state, higher energy level, okay? With fluorescence, let's say, for example, 2-photon fluorescence, what do we have? We have what's called real electronic transitions. Light of the right energy excites, let's say, a fluorophore, so it has, it gets excited to an intermediate, what we call virtual level. Long story, but virtual level is not a real energy level. It's really a superposition of the field with the state of your system itself, okay? But it's quite important in quantum mechanics for explaining multi-photon, for example, processes. So it's an auxiliary level, extremely short-lived. So you have this molecule gets excited to this temporary level, then another photon of the same energy comes in, hits it in the same special mode, so that it excites it to the higher real electronic state. Okay? Now, what happens? So you see all these lines here. Those are vibrational relaxation pathways. So the system is going to lose some energy to the lattice, if you will, right? Usually vis-a-vis some type of heat. And then what will happen is that it relaxes back onto the ground state. Okay? So that means it lost energy. This process we call fluorescence. This is on the order of relaxation to this, about on the order of nanoseconds. Okay? Because it lost energy, the light that's generated doesn't have memory with respect to how it was generated. So we call that process incoherent. As a result, it's omnidirectional and it's unpolarized. Okay? That's fluorescence. The only difference between 2-photon fluorescence versus regular 1-photon fluorescence is that there is no intermediate virtual level. You just come in with one, so instead of, let's say, 2 800 nanometer photons, you come in with 1 400 nanometer photon. And you excite this directly to the higher energy level. And it relaxes, give off fluorescence. That's the only difference between 1 photon and 2 photon. Okay? But it's the workhorse of cellular imaging, right? Now, second harmonic generation, and those based on nonlinear scattering, stimulated Raman scattering, for example, they have, again, ground state looks similar to this one. You excite your system to a higher virtual level. Now, the next photon comes in, same quantum mechanical spatial mode, at the same time. It excites it now to this next virtual level, not the electronic state. But that virtual level is not supposed to be there. It's virtual. And so what happens is an instantaneous relaxation. Okay, so it quickly drops back, and it gives off light that's the sum of the two energies. Right? So now this process has memory. Right? It's coherent. Phase is preserved. So it's highly directional, highly polarized. Key difference. Okay? Key difference that can be exploited. Also, in addition, what happens in comparison to here you're dumping some energy into the system. Here, in principle, you're dumping none. No absorption, just scattering. Remember, scattering is the process that you've been learning about all along. Right? Light bounces off something, right? And there are rules to scattering. This is just scattering non-linearly. So instead of the Chi-1 process, it's the Chi-2 process. With Chi-3, you do something similar, you have third harmonic generation. Keep going higher, fourth, fifth, etc. So what we'll say too is that second harmonic generation could be emitted coherently in the backwards direction under special conditions, right? So generally it's forward facing because of the phase matching condition, right? But if you have scatterers that are spaced, and this really comes from me scattering, that are spaced with an axial period of lambda over 4, where lambda is the second harmonic wavelength, then up to 25% of the signal will be generated in the backscatter direction. Okay? The amount generally that comes in the backwards direction depends on the distribution of scatterers and the tightness of focus in that medium. Okay? But generally, you have backward SHG, because we use it a lot for imaging, simply because you have regular scattering. So some of the light that's propagating in the forward direction hits scatterers and eventually comes in the backscatter direction. Okay, now we think in terms of applying this to a non-linear microscope modality. Okay, that's the process that we're interested in. So to appreciate that process, we must first discuss something, which is the so-called tissue penetration depth, right? So if you want to image tissue, tendon tissue, brain tissue, skin tissue, what have you, there is different attenuations in tissue of light depending on the color of the light, depending on the wavelength. Okay? So if I shine my green laser pointer on my finger, it doesn't light up. Correct? But if I shine my flashlight on my finger, it does light up, right? Well, the green is still getting absorbed. The blue is getting absorbed a lot, right? But the stuff in this pinkish, reddish band, this is what we call the therapeutic window, gets absorbed least. It just scatters around a lot. Right? We call that the therapeutic window. That's the window, if we're interested in really penetrating deeply into tissue, we like to shine light in. Okay? But you all have learned something already about microscopy, I think, right? Right? Yes. Okay. And so you know that there is trade-offs in resolution. There's a diffraction limit. Right? So I want to be in this longer wavelength regime, the so-called therapeutic window, which is about roughly about 700 to about 1200 maybe, 1100 nanometers, right? So that I can penetrate deeply into tissue. But that longer wavelength means lower spatial resolution. Right? That's the nice little paradox with imaging, right? But you want to go to shorter wavelength, and shorter wavelength has a challenge of being absorbed too quickly in the tissue. Not to mention you can kill the tissue, right? And you get to a point where you start to approach mutagenic wavelengths, out to like 350, for example. Okay? So linear microscopy is limited by this problem. It's important to understand that. And the penetration depth effectively winds up being about 50 microns. Nonlinear microscopy, though, has what? This whole thing about frequency conversion that can be exploited. Okay? So a couple of things to note. If you're linear, let's say you're doing linear fluorescence microscopy. So fluorescence is the contrast mechanism. I excite my system with blue light and focus the light down. I'm going to get this nice hourglass shape, hourglass-ish shape through it. So the fluorescence is high here, but you also have a lot of fluorescence bleeding through that you've excited outside of that focal volume. So if you want to discriminate between positions axially along the optical axis, there's a challenge doing that, unless you use confocal microscopy, which is what people use confocal microscopy to do. Okay? Now, with nonlinear microscopy, I talked about getting two photons to interact in the same place in the same time. So we have simultaneity in space and time, right? We're going to get the simultaneity in time by using pulse laser sources, right? Where you have a whole bunch of light photons in a single pulse, but for a very short amount of time so we don't cook the tissue, okay? We're going to get the simultaneity in space by simply focusing tightly. Okay? So now what happens is that when you start to focus your red light through your tissue, no interaction, no interaction, no interaction. Why? Because the probability of getting that multiphoton event is extremely low. I'll talk about what that probability looks like in a second. But it's highest where the intensity of the field is highest, which is exactly at the focus. Right? So that's why you get strong fluorescence here at the focal volume, very little outside of there. What this allows you now to do is optically section. Right? So I'm writing an image by way of, let's say, the fluorescence or even the second harmonic generation. They both operate, if it's multiphoton, the same way. And then what will happen, I'm painting the picture by taking that fluorescence point, moving to the next point, moving to the next point, moving to the next point. Repeat that process, move to the next point, and then build up an image. And I can do that both in X and Y, what we call raster scanning, and then do it along Z. Okay? And now we have my 3D image optically sectioned. The penetration depth goes up by another order of magnitude when you use lasers that are about to a micron. If you go out a little bit further to about 1.3, like a chromium phosphorite laser, you can double that penetration depth. Of course, it also matters the density of the tissue that you're dealing with. Are you trying to image through bone, cornea, skin? So there are other factors that play into it, but roughly you double it, okay? And you get reduced photo damage and no photo bleaching. So Maria Goldberg-Meyer, physicist did this work of multi-photon processes way before the laser was born. She was at U Chicago, and she got a Nobel Prize for completely different work. Right? That's how it used to work. Completely different work. She made a serious contribution to this whole field. And so what she said was, in a typical sunny day, if you have a good 1-photon absorber or a good 2-photon absorber, some specific molecules, the 1-photon absorber would absorb at a rate of about a photon per second. The 2-photon absorber would absorb at a rate of about 1 in every 10 million years. Right? That's a really long time to collect data. Right? So this phenomena cannot be observed until one had very, very, very coherent and intense fields, as what you have with a laser. So when the Ruby laser was born, then they were able to do this kind of cool stuff. Questions so far? Okay. Yep.
I was going to ask, kind of going back to scattering stuff. You mentioned Raman for a second. Is Raman also considered nonlinear?
Yes. No. So, okay. So the stimulated Raman scattering, coherent anti-Stokes Raman scattering. Non-Raman scattering is considered to be, if I'm not mistaken, inelastic scattering, but not necessarily nonlinear. So the nonlinear term here comes in because the intensity of the output, the signal that you measure, is going to depend on the number of photons that are absorbed in that process. Raman uses vibrational excitation modes. When I think of it, this is electronic, right? But you do have the nonlinear equivalent of that with coherent anti-Stokes, and for sure with stimulated Raman scattering. But if I remember correctly, Raman is just a type of inelastic scattering process. Okay. Any other questions? Okay. So here, I just put this up just so you can see it, is that if you have some fluorophores, some fluorescent beads or something, their 1-photon absorption peak is not necessarily the same as 2-photon absorption peak. That has to do with the quantum mechanics. They have different quantum mechanical selection rules. That's all I can really tell you, right? But effectively, for quite a few of these, you'll find that if you can excite them with 1 photon, you can find a wavelength that's much higher to double excite, if you will, with the 2-photon equivalent. And of course, you can also do 3-photon, which people have done now, like Chris Shu out of Cornell, if you have lasers that are at the right wavelengths that are far enough into the mid-IR. Near IR. Okay, like 1.5, 1.6 microns. All right, so I'll just throw this up quickly, because I know you guys may discuss this or may have discussed this. Just in terms of, sometimes it's useful to look at the intensity PSF. Right, so point spread function, you talked about point spread functions, right? So this is obviously a field, the H, rho is the radial coordinate, Z is the axial position, and taking the magnitude squared takes that field and makes it intensity, correct? So we can think of an illumination PSF and a detection PSF when we're thinking about our different microscopy arrangements. So quickly, I throw this up as contrast. For conventional microscopy, you have some source, you have some condenser, and that condenser tries to make the source uniform through the sample, okay? So you get uniform illumination. And then you have an optical imaging system, so-called 4F system, where you relay the information onto a camera, right? It's fast, it's wide field, it's great, works nice. This is a lot of conventional microscopes that we knew about. The imaging here is, the resolution is primarily dictated by the detection PSF. Okay? I mean, this has a strong weighting function with regards to what dictates what the spatial resolution is. And really, spatial resolution, you should always think of it as a 3-dimensional thing. There's X, Y, and Z. We say it all the time, it's a 2D thing, but it's really a 3D thing. So here, you're limited with the axial because everything is flooded, right? The alternative is to have a confocal system, common focus. You all probably know the story about confocal microscopy, right? Marvin Minsky, right? And he just passed maybe 3 weeks ago, I guess, a month ago, something like that. Yeah. And so basically, you have here a pinhole, and you image this pinhole onto your sample. Right? Really clever idea. And then you image that spot. You're going to get fluorescence now only from that small volume because you've restricted it. You blocked it. It's a low-pass filter. You've blocked it everywhere else, right? And now you relay that information onto a point detector, okay, like a PMT. Okay? And huge, huge benefits in imaging. This is now pretty much the workhorse in imaging. When Minsky came up with this, you didn't have the computational power to really do the stitching of all the images together point by point. So the confocal intensity PSF is going to be now what dictates the resolution on both ends. The resolution of this front optical system, what we call the illumination PSF, as well as the resolution of this back optical system, what we call the detection PSF. Now for the 2-photon fluorescence process, here, most of the work is on the front end, right? Because the nature of that 2-photon process is like having something that's intrinsically confocal. Okay? Almost. It doesn't have the same, the confocal pinhole trick is a function of how many area discs, how tightly you focus that pinhole. Okay? But this one is based on this molecular multi-photon process. But because it's a rare process, you've restricted the focal volume intrinsically. Okay? And so what you have here is effectively something that looks confocal at the input, right? But per photon, right? It's per photon 1, per photon 2. So you have the square of these illumination PSFs on both sides dictating the resolution, and then you can have whatever means you want for collection of the signal. Bucket detector, camera, IE, or a point detector. Okay? And yet, you can do one more thing. Most people don't do this. There's a researcher, Stefan Hell, who got the Nobel Prize for one of the people who shared the Nobel Prize a year and a half ago, 2 years ago, for the super resolution microscopy. He's known for doing STED, right? And before he did STED, he worked on something called 4-pi microscopy. And so there, he did 2-photon confocal fluorescence microscopy. And he interfered it, right? So it was ridiculous. I was a postdoc working on this. It's not a trivial experiment. Anyway, so what happens in that case is that in addition to having an intrinsic confocality for the 2-photon process, you actually have a confocal pinhole at the detection end. This is very good for certain signal processing effects that are not clear from the short lecture I'm giving you. So essentially, what you're trying to do is squeeze and squeeze and squeeze this intensity PSF. But that comes at a cost. Why do you want to do that? Because you want to get a very nice sharp image. And a nice sharp image means smaller spot, which means smaller point, right? You don't want your point to be too spread through the system, right? So this is a delta function in 2 dimensions, right? So point spread function. So what happens though, when you start to squeeze, 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 you start to get additional side lobes, intensity functions, and that same distribution that start to increase, right? And that can interfere the main PSF that you want. And Hell figured out a way to suppress enough those side lobes by using an additional confocal pinhole. You got beautiful resolution, about a sub 100 nanometers using 800 nanometer wavelength excitation. Axially. That's impressive. All right, quickly, the setup is as follows. Okay, this might take a little time. So let's just quickly say that the 1-photon, the 2-photon case looks similar. The difference with the 2-photon case is that you're really making up an image point by point. The scanner tries to do that. You put in colored filters here. colored beam splitters, so-called dichroic filters, so that you transmit long wavelengths and you reflect short wavelengths into a detector. Okay, that's the story. You may have questions about exactly what the resolution is. Spatial resolution, I'll give you here this chart of two-photon fluorescence and SHG versus conventional and confocal microscopy. If you plug some numbers in, you'll find that the SHG and the two-photon have, for these numbers, have resolutions that are not that great in comparison to conventional and confocal, right? Not that great, but they're really, really useful, because there's one thing here that's that really underscores the importance of the techniques, which is one, penetration depth, which is not shown on this chart, right? So this is comparing these imaging techniques for longer wavelengths is what you use for the multiphoton case. Shorter wavelengths is what you use for the other cases, right? And the potential for photo damage. So you want to be able, so they really gain their strength from being able to penetrate deeply into tissue with ideally hardly damage in it, okay? And on top of that, as we'll note in a second, second harmonic, you don't need fluorophores. So it has an intrinsic contrast mechanism that's unique to the particular biological structure. If it's Chi2, it will automatically give you a second harmonic signal. Okay, in terms of setup, they're both pricey. SSG is generally label free and less heat into the specimen, as I mentioned. So I'll quickly go through some things that we've done in my group. We've looked at tendon injury, and specifically we focus on horse tendon. We've collaborated with, I don't know if you know, Dr. Shiv in IGB, as well as some people in Vet Med a few years ago. And what we found was that they wanted to look at horse tendon, right? And a horse that was considered, the term as an equine athlete. That was new for me. So basically a racehorse. And when the horse overuses his tendon, it gets tendinitis, right? So it's like the tennis elbow thing that we can get. And so what happens is that these researchers were interested in stem cell regeneration of damaged fibers, and they wanted to look at the collagen fiber distribution in the tissue. Collagen is a protein that's chiral, okay? It's a triple helix structure, and when you have enough of this collagen molecule, it can give off a second harmonic signal. They use polarized light microscopy to see this, but the polarization structure here, the contrast that was there was still not high enough, so they added a dye to exaggerate the contrast. Contrast difference between ordinary and extraordinary index of refraction. Okay, and these are the images that they get. Normal tendon, injured tendon. Now when you apply your second harmonic to this, without the dye, it's automatically clear what the differences in the images are. These fibers all align in the same direction, right? They have some biomechanical function with regards to dealing with an applied load. Here the fibers are disrupted. This clarity in the image could allow you to do some type of image analysis and where you can do something quantitative with that. You can also apply this to bone. Bone tissue is nothing more than mineralized collagen. So it's collagen with minerals on it. That's what gives it its hardness. And here, these biomechanicians, they typically use scanning electron microscopy to image bone, and this is an SEM image, but they don't have the specificity. Right? So you can combine that, or instead use second harmonic, and you can see here, all we are seeing are the collagen fibers. And what we have, these are called Harvesian canals, these dark voids. You can do this as a function of age in particular. And what you also have are these almost like rugs, right? So these are collagen fibers, they're rope-like structures, these proteins, and they can form what's called lamella sheets, where they lay in a bed with respect to each other. Again, that has a certain biomechanical function, and they can even wrap around certain features in a biological tissue, and that's when they do that here, we call them Harvesian canals. Okay? And you can see all that automatically without labeling, with not bad spatial resolution. You can look at the cervical microenvironment and try to determine something about its structure of how the collagen fibers in that cervical microenvironment are arranged, right? This is important for studies on preterm birth. The contrast is ironically enough not great here. I should have gone darker, but that's fine. So here, what you should see is a ring of fibers. Okay, so this is rat cervix. There's a canal. The difference with human cervix is that this rat cervix has actually two canals. There's a thin sheet of collagen in here, and then you have these circumferential fibers that wrap around. You can study something about the distribution of these fibers. Let's say using spatial frequency analysis, taking it to the Fourier domain, for example, which is something we do, spatial frequencies, right? And here, what's fascinating, because since the SHG is 3D, is that you can get a volumetric image. And then you can try to study now these fiber distributions. So here, what we're finding out, if we take two regions of interest, 1 and 2, apply some analysis, everything in blue, purple is telling us these are fibers that are oriented in this structure, in this plane, in some direction along X and Y. Everything in red is telling they're fibers that snake out of a plane and are projected along the Z. You cannot do that, or you cannot determine that easily unless you have a method of doing 3D sectioning. And that's what we saw here. Okay. You can also, I mentioned the whole thing about the polarization information is technically in there. So P and E here are no longer parallel. They're not parallel vectors. Right? In general, when you have an anisotropic medium, P is going to be some weighted superposition of the components of E. Right? And P itself will have components, correct? And those weights are going to be, or can be represented as tensor elements. So these D's are related to the chi. Okay? What we do is we assume a certain crystallographic symmetry, something, for example, like hexagonal. It makes this matrix sparse, lots of zeros everywhere. We normalize with respect to some of these elements, and we write down an intensity expression for the second harmonic that's dependent on the polarization angle of the input field. That's what we call alpha, right? And you get a distribution. Right, so this is our polarization signature as a function of angle. It's normalized. And we create an artificial scaffold of collagen in the lab. We analyze that tissue. This now is subdiffraction information. We're looking at how light is scattered by the molecular arrangements themselves. Right? We don't know exactly what that means. But what that does is allows us to try to get some estimate of these normalized coefficients. And we want to use that as an additional means for differentiating with tissue types. Right? You can do the same thing with the sclera and the cornea.
What do those signals actually mean?
So remember now, so here, they tell you something about how light scattered off of it. Right? Because the polarization density tells you something about the dipole moments. But the polarization for the medium to be anisotropic, meaning that it's polarization dependent, you have components of that polarization density. And each component of the polarization density is related to each component of the electric field by some weight. What we do is assume a certain type of structure so that we make a lot of these zeros. And we normalize, in this case with respect to the D15, and we write down an intensity expression for the second harmonic light as a function of the polarization angle with coefficients that correspond to how the light scattered. Right? And we try to use that as some metric for assessing the tissues that we image. Right? And so all we did here was we got some estimate of what these Ds look like for a collagen scaffold that you can engineer in the lab. So tissue engineers do this all the time. And we looked at, I think this was pig. The sclera is the white part of your eye, which is collagen. And the cornea is the transparent part of your eye, right? And so we're seeing here that the collagen information, as far as we can tell with our SHG technique, is the same. The arrangement is different, which lends itself to why there's opacity and transparency for these two different parts of the eye, along with hydration as well. Okay? So this was just another way of trying to exploit the polarization information to see if we can get something other than just what we see in the picture.
Do you think those numbers change when you have excised ex vivo tissue versus when it's just fresh? So
that's a whole another very interesting story that no one's really done a controlled experiment with yet. There's a lot of assumptions. Many biologists would tell you they expect change. They try to do something that is a good enough approximation to being in the whole system, having the right temperature, having the right buffer solution. But it depends on what type of biology you're studying, right? But they do it all the time. Right? Now, there are other problems with this that I can talk to you offline about. But the idea we were exploring was whether or not these numbers would change as some function of some type of molecular change in the collagen. And to do that, we looked at breast biopsies as a function of different pathologies. And we looked specifically at normal breast biopsy and compared it to something that looks like scar tissue, hyperplastic, something that looks precancerous, called dysplasia, and something that was actually malignant, right? And here, we did a study a few years ago using those parameters, and we got some promising results that suggested that we can do some level of differentiation between some of those pathologies based on changes in those numbers. Okay, and again, I'll talk to you offline about what the limitations of that are. So finally, so we have the SHG. It provides intrinsic contrast for biological tissues. You can do some interesting quantitative things with this technique. You can exploit the polarization degree of freedom. One other thing that we've been doing recently, again, sorry for the contrast, is that can you?
Yeah.
Thanks, just for a moment. There we go. Is that we've been applying this also to breast biopsies at different stages, looking at third harmonic generation in combination with second harmonic generation, and where the THG is in purple and the green is SHG, and all you're doing is imaging through the tissue section in volume, and then you stack that all up, create a so-called Z-stack, and you play the movie, right? And that's what the green and that does. And this is a tiled image doing the same thing through another region in breast biopsy. Okay. And that, I think, is all. Thanks. Now, questions? I'm going to make the slides available to you guys. I'll email Professor Popescu so that you have it. I saw the email about you guys potentially being tested on that, right? So I'll send it to you. Any questions?
So I've heard about in nonlinear there's something about self-focusing.
Oh, yeah, I didn't mention that. Sure, yeah, sure. So what happens is that, so let's say if you have a medium that's chi3, right? You can have something that's called the optical Kerr effect, right? So you have an index of refraction that is dependent on the intensity. So n changes as a function of I. Now, if that change is in the transverse plane, xy, and if you're sitting in a Gaussian beam, then the index is going to be high on axis, where you have the highest intensity, low off axis, where you have the lowest intensity. And it would actually act like a lens. So it would actually act like a graded index material, a Grin lens. That's called self-focusing. So there are all kinds of interesting phenomena that can happen with nonlinearities. That can be quite complicated, as a matter of fact. Soliton waves used to be a big thing when I was a student. It was tries to exploit certain types of nonlinearities so you can have a perfect pulse traveling almost indefinitely through a medium.
So what kind of intensities do you need to get SAG traveling?
It depends on the tissue. I'm used to thinking here in terms of power. So if we use something like a 1.2, as a matter of fact, if you use like even a 0.8 numerical aperture lens, so the lenses are characterized by how strongly they focus, and numerical aperture is one way of doing that. And if it's something like tendon tissue, which has a lot of collagen fibers, you can come in with just a few milliwatts, 2, 3, 5 milliwatts of average power of 800 nanometer light, right? And you will get a decent signal. The second harmonic signal, though, is a much rarer process than fluorescence. So fluorescence is at least 2 or 3 orders of magnitude more intense than second harmonic. The strength of the second harmonic, especially now, is that you have a lot of people in biology who are interested in mechanobiology. They're interested in the extracellular environment. What's this? These cells are nested in stuff. Turns out they're collagen fibers. And they exert forces on each other vis-a-vis these collagen highways. And people are very interested in, ideally, without disturbing the biological system via labeling, because you're still excising the tissue, to image and quantify and understand what's going on in the extracellular matrix. And so if you have a means of doing that with something like a second harmonic, then it's helpful. It would even be more helpful if you can, so I didn't tell you, but there's like 20 different types of collagen, and they're called different types of collagen. And so the ones that we focus on is type 1, which is very prevalent because type 1 is highly crystalline and it gives the strongest second harmonic signal. Not all of them are going to give a second harmonic signal. Type 1, I think type 2, type 3, they do. Type 5, I think, does. But not all of them.
So there's no risk of like damaging?
So I didn't put a slide in here. So what happens is there's a sweet spot. And so we work with 100 femtosecond pulse width. So if you're and the rep rate, so the distance between pulses is going to be on the order of 80 megahertz. So if you have pulse widths that are somewhere between 80 femtoseconds to about 150 femtoseconds, you're right about a sweet spot where you can get enough energy, enough of this 2-photon effect to see a good second harmonic signal. However, if you start to move to really, really, really short pulses, 10 femtoseconds, for example, then what happens is you start to worry about creating sort of these multi-impact ionization effects. So you're going to get a second harmonic, but you're also going to now excite other processes, which could be very damaging in the system. If you work the other way around, and I was a postdoc, and I remember there were researchers who bought a commercial multi-photon system, and they could not get it to work for the longest time. And it was because it had to do with patent laws. But effectively, when they bought the system, there was a block of glass in the laser. And that block of glass was adding material dispersion, so it was spreading the pulse. So what happened, it was like shining a CW laser on the tissue. So you'll get, eventually, a multi-photon effect, but you're coming in with at least 100 milliwatts of power, if not much more, to get a signal. And then now you're also cooking the sample. So that's why there's a sweet spot where you just need these short pulses for short durations, but they have a lot of photons in them to get the temporal crowding effect. But yes, otherwise you don't destroy anything. And there's tons of people who've done stuff in vitro, monitoring of, let's say, zebrafish, and looking at development of neural tissue over, at least over a couple of days. Any other questions? Good. Well, thank you. Appreciate it.